Have you ever wondered how crucial the role of participant selection is in drug development trials? Well, it's like choosing the right ingredients for a recipe. Just as the wrong ingredient can spoil the dish, a poorly chosen participant can skew the results of a drug trial, leading to inaccurate conclusions. Choosing the right participants for a study is not just about the numbers. It's about ensuring that the selected individuals accurately represent the population that the drug is intended for. This involves a careful consideration of factors such as age, gender, health status, and even genetic makeup. Imagine you're developing a drug for a condition that primarily affects elderly women. If your study only includes young, healthy men, your results won't be applicable to your target population. This could lead to ineffective treatments or even harmful side effects when the drug is released. As you can see, the selection of participants is an intricate process that plays a significant role in the success of drug development trials. Now, you might be curious about how researchers go about selecting participants for these trials. At the heart of any trial is the participant pool. The size of this pool is crucial. Researchers aim for a count that's not too large to keep the study manageable and cost-effective, and not too small to ensure statistical significance. This balance is struck based on the anticipated effect of the drug and the variability in the population. Next comes the process of inclusion, where eligible participants are identified. This is where researchers define the characteristics a patient must display to participate. These characteristics, or inclusion criteria, can range from age and gender to specific health conditions, or even lifestyle factors. These are set to ensure the results can be attributed to the drug being tested and not confounded by other variables. However, not everyone who fits the inclusion criteria can participate. There are always conditions that render a patient ineligible. These exclusion criteria could be based on factors like severe health conditions, potential drug interactions, or even ethical considerations, such as pregnancy or inability to give informed consent. They are put in place to protect participants and maintain the integrity of the trial. This selection process ensures a balance between obtaining robust data and maintaining ethical standards. Researchers meticulously justify their selection criteria, always aiming for a representative and safe participant pool. Through this rigorous process, researchers strive to ensure that their study's results will be valid and reliable. To wrap up, let's summarize the key points about participant selection in drug development. The selection of participants is a critical step in the process of drug development. It's not just about numbers, but about the right kind of participants that can provide meaningful data. The desired number of participants is not plucked out of thin air. Rather, it is determined by several factors, including the nature of the drug, the disease it's intended to treat, and the phase of the trial. A larger number might be needed for diseases with a high variation in symptoms, while a smaller group could suffice for more specific conditions. The inclusion criteria that researchers set is not arbitrary either. It is designed to ensure that the participants have the characteristics that are relevant to the study. For example, if the drug is intended for a certain age group or a specific medical condition, then those are the participants that need to be included. On the flip side, there are also conditions that make a patient ineligible to participate. These exclusion criteria are put in place to protect the safety of the participants and the integrity of the data. For instance, individuals with certain health conditions might be excluded if participating could potentially harm them or skew the data. Remember, the success of drug development trials hinges on the careful selection of participants. It's a meticulous process, but one that is crucial to the advancement of medical science.